Hello friends, today we are going to study different structures of proteins. There are four structures of protein: primary structure, secondary structure, tertiary structure and quaternary structure. So we are going to study theory part of those structure today. So let's start with primary structure of proteins. Primary structure of proteins is the sequence of constituent alpha amino acid residues linked by peptide bond. Any change in the sequence of amino acid residues result in a different protein. Primary structure of proteins is represented by writing the three-letter symbols of amino acid residues as per their sequence in the concerned protein. The symbols are separated by dashes. According to the convention, the N-terminal amino acid residue is written at the left end and the C-terminal amino acid residue at the right end. So that's all about primary structure of amino acid. Now let's see the theory of secondary structure of amino acid. Secondary structure of let's see the secondary structure of proteins. Secondary structure of proteins, the three-dimensional arrangement of localized regions of a protein chain is called the secondary structure of proteins. Hydrogen bonding between NH proton of one amide linkage and C double bond O oxygen of another give rise to the secondary structure. Two types of secondary structures commonly found in proteins are alpha helix and beta pleated sheet. Now let's discuss alpha helix and beta pleated sheet structures. Alpha helix. The alpha helix forms when a polypeptide chain twists into a right-handed or clockwise spiral. Some characteristic features of alpha helical structure of proteins are: each turn of alpha helix has 3.6 amino acids. A C double bond O group of one amino acid is hydrogen bonded to NH group of the fourth amino acid along the chain. Hydrogen bonds are parallel to the axis of helix while R groups extend outwards from the helices. They are outward from the helix core. Example of alpha helix is myosin in muscle and alpha keratin in hair are proteins with almost entire alpha helical secondary structure. Now let's start with next second part of secondary structure of protein. Second type, beta pleated sheet. The secondary structure is called beta pleated sheet when two or more polypeptide chains are called strands line up side by side. The beta pleated sheet structure of protein consists of extended strands of polypeptide chains held together by hydrogen bonding. The characteristic of beta pleated sheet structures are: the C double bond O and NH bonds lie in the planes of the sheet. Hydrogen bonding occurs between the NH and C double bond O groups of nearby amino acid residues in the neighboring chains. Third point, the R groups are oriented above and below the plane of the sheets. Fourth, beta pleated sheet arrangement is favored by amino acid with small R groups. So here we completed with beta pleated sheet. Secondary structure of protein is finished. Now let's go to tertiary structure of protein. It is the result of folding of the chain in a particular manner that the structure is itself stabilized and also has attractive interaction with the aqueous environment of the cell. The globular and fibrous proteins represent two major molecular shapes resulting from the tertiary structure. We distinguished globular and fibrous protein in earlier video. The forces that stabilize a particular tertiary structure include hydrogen bonding, dipole-dipole attraction, that is due to polar bond in the side chain, electrostatic attraction due to ionic groups like COO-, NH3+ in the side chain, and also London dispersion forces, which are also called van der Waals forces. Fourth point, finally disulfide bonds formed by oxidation of nearby SH groups in cysteine residues are the covalent bonds which stabilize the tertiary structure. So here tertiary structure is finished, and now let's go to the theory part of quaternary structures of proteins. When two or more polypeptide chains with folded tertiary structures come together in one protein complex, the resulting shape is called quaternary structure of the protein. Each individual polypeptide chain is called a subunit of the overall protein. For example, hemoglobin consists of four subunits called heme held together by intermolecular forces in a compact three-dimensional shape. Hemoglobin can do its function of oxygen transport only when all the four subunits are together. So that's all about quaternary structure of proteins. So we discuss all four types of protein: primary, secondary, tertiary, and quaternary in detail, the theory parts. Thanks for watching the video. Bye.